Welcome dear students, this is the fourth part of my cell cycle lecture. In my first class, I have discussed about the basic conception about cell cycle. In the second class, I have discussed about how G1S transition occur. This is called first checkpoint during this cell cycle. This is also known as cell cycle regulations. And in the third part of my lecture, I have discussed about how replication are regulated during this S phase so that replication occurs once and only once per cell cycle. Now in this class I will talk about how M phase CDK, that means CDK1, are regulated so that only at this M phase this CDK molecule can function. In my first class I have mentioned that the CDK molecules are always there. The CDK molecules for M phase are always present throughout the cell cycle. It present in the G1, S phase, G2, M phase, always present. But the CDK molecules that perform in during this M phase called CDK1, the CDK1 molecule is present throughout the cell cycle, but it is not active in condition throughout the cell cycle, only gets activated during this M phase. And the example is like G1 phase CDK, like CDK4, CDK6 are present but are not activated all throughout this cell cycle, only activated during this G1 phase. And how this occurs? Because this occurs by the association with cyclin molecules. So M phase cyclins called cyclin A or B is important for the activation of CDK1, and the cyclin actually is not present throughout the cell cycle. It starts to accumulate from the S phase throughout the G2 and ultimately to the M phase. So that's why CDK1 is not active in conditions. So cyclin A or B, this cyclin molecule starts to accumulate from the S phase throughout the G2 and M phase, then it associates with the CDK1. But during this S phase, when the cyclin A or cyclin B are present, then also the CDK1 that calls the M phase CDK is not active during this S phase. I am repeating again. The cyclin for the M CDK, that means M cyclin, this is the M cyclin and this M CDK. This M cyclin or mitotic cyclin, M phase cyclin, starts to produce or accumulate from the S phase. And I have mentioned that M CDK is present throughout the cell cycle. But why then in the S phase this CDK1 is not activated? Only it has been found that during this M phase this CDK1 gets activated. The mechanism behind this is phosphorylations. In the CDK there are tyrosine at the 15 position and threonine at the 14 positions that get phosphorylated. These two amino acids get phosphorylated by an enzyme called WEE1. This is a kinase enzyme that can phosphorylate this CDK1, and when this CDK1 gets phosphorylated at this particular amino acid residue, it is inactive in conditions. It is inactive in conditions. Remain inactive. Okay. Now there is a counterbalance of this kinase that is called CDC25 family of protein, specifically CDC25C protein, that is actually phosphatase. And this protein, actually enzyme, can dephosphorylate these two proteins. So what happens then? The cyclin A or B is associated with a CDK1 that have the tyrosine 15 and threonine 14 but not phosphorylated in conditions, and in that state we call this M CDK, M phase CDK, active in condition. Okay, it is inactive in condition, it is active in condition. So there is a counterbalance. Now what happens, what triggers the CDC25 to dephosphorylate these two amino acids. It has been found that during this S phase, from the S phase, late S phase, this molecule starts to active, and this molecule remove this phosphate group. And another important fact is this active MCDK helps in the activation of this CDC25. This is called positive loop. How this occur? Because this CDK actually phosphorylate this CDC25C, and unless this CDC25C gets phosphorylated, it cannot dephosphorylate this molecule. So this is the positive loop. But another important thing is this molecule, this same active M phase CDK, simultaneously inhibit V1 kinase. This is also a positive loop, because by inhibition it actually inhibits these phosphorylations. So it inhibits the inactivation of this M phase CDK. Okay, so there is a relationship of this molecule. Okay, another is during this G2 phase, when the cell size actually is accurate, then a protein actually gets activated called CDR2. When the cell size is just enough to divide, then a protein activates called CDR2. The CDR2 protein inhibits V1 kinase. So now you can see here that this V1 actually inhibits this process from these activations, but V1 gets inactivated by the CDR2 and also by active M phase CDK. By this way, so if I draw a graph, you can see that this is the S phase, G2 and this M phase. The cyclin of this M phase cyclin, like cyclin A or B, actually starts to produce from this S phase throughout the G2 to the M phase. But the activation of the CDK happens only during this M phase. The peak occurs in the M phase only, and this is the reason, because in the S phase the CDC25 is not activated, but after that CDC25 starts to activate it, and that's why the CDK becomes dephosphorylated and becomes active. So this is all about regulation of M phase CDK. Hope this video will help you. Thank you for watching, and always keep learning.